Welcome everyone to the AstroAI lunch talk. Today, I am very happy to introduce Zachary. Zach is a fourth-year grad student at MIT working on Brett Maguire's lab. He is a chemist, which is not very common in AstroAI talks, but we're very happy to have more of you coming. And he works on combining computational methods with astronomical observations to find molecules in space. Last year, he was awarded the Rao Prize in an interesting and prestigious molecular spectroscopy symposium. And today, he's going to be telling us about automating assignment of interstellar line surveys, presumably, as I always tend to suppose, using machine learning. And if I understand correctly, it has to do something. It has to do with something like replacing human intuition with some sort of system for this sort of identification. So looking forward to your talk. Floor is yours. You're welcome to interact with questions anytime, and you have potentially water. So go for it.
Yeah, thank you for that introduction. And thank you for having me out. Today, I'm excited to present some recent work I've done on automating the assignment of interstellar line surveys via radio astronomical observations. So just to set a little bit of the context, as astrochemists, we really care about understanding and analyzing interstellar molecules. But why is this? What information can we really gauge from this process? Well, as all of you astronomers know, because we're looking all the way out in space, we're not fortunate to, say, use a thermometer to gauge temperature and things like that. Instead, in order to trace the physical properties and evolutionary history of interstellar sources, we oftentimes rely on the molecules we detect, as well as the properties of those molecules. Just a couple of examples, briefly. Deuterium ratios can provide information about the temperature during the formation of a source, and detecting a molecule like SiO can pretty sensitively trace things like stellar outflows or interstellar shocks. So if an astrochemist wants to decipher a molecular inventory of an interstellar source, or try to detect a new molecule in space, oftentimes we rely on radio astronomical observations. And the project that I'll be presenting today is mainly focused on automating the assignment of these. So building some algorithm that can automatically tell you which molecules are present in this data. But before I get into that, just to talk a little bit more about these observations, what you can see on the screen is the line survey of a certain star forming region that was collected using the ALMA array. And generally, most of the lines that you see in this spectrum are rotational emission of molecules in the gas phase in this interstellar source. So in theory, if we wanted to manually go ahead and determine which molecules are present in this line survey, we could go to a rotational spectroscopy database and see or try to cross-reference the frequencies in the database with the frequencies we see in the spectrum, and from that, try to figure out which molecules are present in the data. But as I'll go on to show in the next couple of slides, at least manually, this can be pretty challenging. The first reason for this is that these line surveys can be just extremely dense. Here I've just zoomed in on a narrow frequency region of the observations, and you can see there's tons and tons of lines here. I mean, over the entire broadband frequency range, there's well over 1,000 different transitions. So if someone were to manually, for all of these lines, go to a rotational spectroscopy database, cross-reference the spectra, see if it lines up, it would take probably weeks of pretty painstaking effort and is not a great use of PhD time. Additionally, even if we want to go ahead and manually assign all these lines based on this spectroscopy, doing this can be quite challenging. And to show this, I've just zoomed in on a single line from a rotational spectrum here. So say that we see this line and we want to go and figure out what the molecular carrier is. So we go to, as I mentioned, a rotational spectroscopy database, and the first thing that we probably see is that ace-naphthalene, the molecule I've shown here on the screen, has a known transition that lines up almost perfectly with the center of the observed peak. So that's great. We can go ahead and assign this line to ace-naphthalene and move on to the next line in our spectrum, right? But well, not so fast, because if we keep looking in the database, we'll end up finding that there are 6 different known molecules with known transitions that are right nearby this observed peak. In fact, in every single case, the uncertainty of the catalog transition actually overlaps with the center frequency of the observed peak. As I mentioned previously, molecule C, ace-naphthalene, has the closest frequency match in the database to the observed line.
Sorry.
Yeah.
So are your database sort of theoretical? Okay.
Sorry, yeah, go ahead, go
I
ahead.
I was gonna ask if those uncertainties from the catalog, from the, sorry, database are usually larger than the actual observational uncertainty on the position of the line.
So that very much depends on the experiment. So these databases are experimental rotational spectra that were collected. So all of these lines are known rotational transitions of these 6 molecules, and the uncertainty depends on the experiment. It depends on the experimental uncertainty during the experiment, as well as how far we're extrapolating from the fit there. Yeah. Yes, wrong way, I think. Yes. So that's just pretty much the intensity unit that is used for a lot of these radioastronomical observations. It's used oftentimes just because it allows us to normalize like black body radiation with changes in temperature over the frequency range. So that's the general intensity unit that you can use these radioastronomical observations. Yeah. Okay, so yeah, as I mentioned, molecule C here, asynaptylene, has an almost perfect frequency match. However, through further investigation of things like the line intensities and whatnot, we actually know that molecule D is the correct molecular carrier of this line. So this just really goes to show that assigning these spectra can be pretty challenging based on the spectroscopic match alone. It can be a pretty confusing process to go about. So it'd be really nice, therefore, if we had some additional information that we could use to really automatically or accurately assign these lines to specific molecular carriers, just to make sure that we're assigning the correct molecular inventory in these interstellar sources. And one piece of information that we can use here is the fact that oftentimes in these interstellar sources, the detected abundant molecules are generally quite chemically related. And that's just because due to the unique conditions in these different interstellar sources, different chemistry can actually occur. As a result, different molecules can be put into the gas phase where they can be detected by our radio telescope. So what I've shown here is some of the most abundant molecules detected toward the TMC1 cold dark cloud. And this is a molecular cloud source that's prior to the star formation process. So the temperatures are super cold, around like 10 Kelvin. And visually, you can see that a lot of these molecules look pretty similar. They're mainly these carbon chain species that are really unsaturated, meaning that they have lots of triple or double bonds. But now, on this next slide, if you go to the most abundant molecules detected toward IRAS 16293b, which is a star-forming region, so a source in which a star is actually in the process of forming, the temperatures are way, way warmer, up to hundreds of Kelvin. You can see that the molecules on this side look pretty different than those on the last slide. Here you have mainly these oxygen-containing species that are really saturated, so they have lots of these single bonds instead. In fact, looking a little bit closer, all of these molecules have carbonyl groups, all of these ones have OH or hydroxyl groups, and all of these ones have CH3O or methoxy groups. So this just goes to show that in these interstellar sources, the molecular inventory can be quite homogeneous. It's generally fairly homogeneous, at least within individual sources. However, between different sources, especially those of different source types, like a molecular cloud to a star-forming region, oftentimes the chemical inventories are quite unique. So with this in mind, it would be really nice if we could incorporate this logic into our line assignment algorithm. So we're not only reliant on looking at the spectroscopic match, which as I showed a couple slides ago can be a little bit tenuous, but we also have a way that for every single molecular candidate for a line that we want to assign, we can evaluate its structural or chemical relevance based on what we know about the source. And in the past, if we wanted to do this manually, generally we relied on chemical intuition. There was a scientist saying, well, I've seen these molecules before, this molecule looks pretty similar, so we can assign it. Or this molecule is really weird, we wouldn't expect it to be there. But that's a hard thing to automate because it's really just chemical intuition that you build up as a chemist by looking at way too many molecules during the course of your PhD. So this is something that we're going to try to automate during this process. And the way that we're going to go about doing this, as we'll get into the details in the next couple of slides, is during the assignment process, what we're going to really try to do is build up a chemical space surface with weight concentrated around the known detected molecules in this source. So we'll have weight in the regions of space occupied by the molecules that we know are in this source. And because similar molecules will also, in theory, be nearby one another in this chemical space, we'll be able to use this as a heuristic to determine for every new candidate how likely it is or how chemically relevant it is to the interstellar source. So in order to do this, the first thing that we need is some sort of embedding model to turn our molecules into some machine-readable vector form. And for this, we've been utilizing this VicGay model. And this is a model that was developed by Dr. Kelvin Lee, who's a former postdoc here at our group at MIT, but also here at the CFA, where he was a postdoc as well. He's now working over at Intel. But he developed this astrochemistry-focused embedding model, which works on selfie string representations of molecules. So these selfie strings, as you can see here on the left side of the string, are tokenized string representations of molecular subunits. So you can see here for glycolaldehyde, the molecule shown on the left side of the screen, the selfie string, the first token is that oxygen, the second one is the double bonded C, followed by the subsequent carbon, and followed by the single bonded oxygen. So that's how you build up a selfie string representation of a molecule. Ultimately, the model architecture is the sequence-to-sequence model, which has this encoder, followed by a decoder, each of which are individual GRU subunits. And how it was trained was Kelvin amassed this large data set. Yes, go ahead.
Sorry,
Yeah.
could you clarify for those who are non-experts what a GRU is?
Exactly.
Yeah.
So it's the sequence-to-sequence model. It's pretty much GRUs are pretty much like newer versions of recurrent neural networks, which work on sequences of, say, it's really good for this purpose where you have these sequences of tokens, where it's a model that sequentially processes each token and passes on the new information combined from all of the previous tokens as it sequences the entire sequence one by one in each of these units. GRUs are really nice because an issue with recurrent neural networks is they had this sort of like short-term memory issue due to the vanishing gradient problem, where if you have a long sequence, it really had short-term memory that by the time that you're encoding or passing on information for this entire sequence, it would kind of forget what it had seen earlier on in the sequence. So it had this sort of short-term memory problem. GRUs fix this by adding gates to this process where it can select which information is passed on and which information is discarded. So it pretty much only passes on the important information and discards kind of the junk. And as a result, it's able to fix this vanishing gradient or this like short-term memory problem. So it's able to really efficiently pass on the information from each step of the sequence. That anything else? Cool. All right, perfect. Perfect. So yeah, how this model was trained was Kelvin amassed, and I don't want to take any credit for training this model. This was all Kelvin's work, but he amassed a very large data set of astrochemically relevant molecules. And what he did is he got them in their selfie string representation and then randomly masked out some selfies tokens. Then by passing it to the encoder and the decoder, what the model was trained to do was ultimately predict correctly the masked token. So what it requires then is for this encoder to be able to learn how to embed these selfies tokens in really information dense and contextually aware representations. So ultimately, now that the model was trained, and it's available open source on Kelvin's GitHub page, what we can do is take any new molecule, run it through this embedding model, and what it will do is for every selfies token, take its embedding representation from the encoder, combine them to create these 32-dimensional dense vector representations of molecules. So ultimately, it creates similar vector representations for molecules that are structurally or chemically similar is the general idea here. So now that we have our embedding model, we want to go ahead. Yes, go ahead.
Before you move on.
Yes.
So you said things like tokenization and next token prediction or in between token. So and that's the sort of things that large language models do using transformers. Was there a reason why a GRU was used here instead of a transformer architecture?
That's a great question that I don't have the answer for. As I said, I'm just using this Astrochem specific embedder, but was not actually who developed it, so I apologize.
No.
I have the own answer there.
Maybe it would be interesting to see if a transformer could do a better job
Yeah.
at creating representations that are more
Yeah, no, absolutely. I know that making like meaningful molecular embeddings is something that's super important for chemistry related machine learning. So it's a very active field of study. Uh, yeah, thanks. Yeah. Okay, so now that we have our embedding model, what we want to do is, as I mentioned, we want to build up this chemical space surface to understand the regions of chemical space occupied by these molecules as we're sort of going through the assignment process. So here, what I've shown is all of the molecules detected toward that IRS 16293b star-forming region, and I've taken their Wittge vector representations and just U-mapped them to 2 dimensions, and this is purely just for visualization purposes, and I'm just showing them on this figure here for reference. But say that we're at a point where we're just starting our algorithm, we haven't yet assigned a single line in our data, and therefore we in theory have no understanding or no knowledge of the chemical inventory of the source. As a result, at this point, you can see that the space surface is completely flat. It's 0 at all points. But how this algorithm works is it's sequential, starting with the strongest line in the spectrum. So the first thing that we do is we go ahead and try to assign that strongest line in the spectrum based on the spectroscopic match alone. And we know for this data set that first strongest line in the spectrum corresponds to a transition of formaldehyde. And I put now a star at that region in chemical space relating to this molecule. And now that we know that that molecule is in our data, we know that it's present in our interstellar source, we start by building up weight on the chemical space surface at the point related to that molecule. And that's because now that we know that that molecule is present in our data, it makes sense now for chemically similar molecules to also be present. So we're starting to build up weight on the surface based on what we know about the source. We then go on to the next strongest line. We know it's CH3CHO, continue to build up weight on this chemical space surface and go molecule by molecule. As we assign new molecules, we're continuing to build up a more informed understanding of this chemical space surface. So we're at a point now, which I've just flipped through, where we've assigned only 5 molecules in this spectrum. And as we saw previously, there's thousands of different lines in this line survey, yet we've only analyzed or investigated the first handful, the first small number of them. And you can see at this point, all of the molecules that we'll ultimately want to assign, even as we get to the weakest lines in the spectrum, are already in highly weighted regions of chemical space. Therefore, we can use this weight surface as sort of a heuristic to determine how relevant or how chemically similar any new molecular candidate is to this interstellar source. And just to show how this works a little bit more, here I've put a purple square at the locations in chemical space corresponding to methylformate, so that's that oxygen-bearing molecule, CH3OCHO, as well as HC9N and indene. And methylformate is a molecule that's known to be detected in IRS 16293b, although it's not corresponding to one of the 5 strongest molecules that we've already assigned at this point. On the other hand, HC9N and indene are molecules that we know to be present in a cold molecular cloud like TMC1. However, we likely would not detect it in a source like IRS 16293b. You can see, therefore, that methylformate is clearly in a much more highly weighted region of chemical space, as opposed to HC9N and indene, where it's pretty much flat at those points. So as a result, if for whatever reason we came about a line in which the molecular candidates for that line based on the spectroscopy alone is, say, methylformate and then HC9N, we'll say that we can't differentiate them based on the spectroscopy alone because, as I showed a couple of slides ago, this can be a little bit challenging or tricky. Well, this weight surface will allow us to really understand and really quantify how chemically relevant or chemically similar each of these molecules are based on what we know about the source. And as a result, in this case, methylformate would be the molecule that's detected. So just to walk through how the entire algorithm works, the first thing that we obviously need is a line survey collected toward our interstellar source. We then run a peak finder to determine the peak frequencies and intensities. And starting with the strongest line in the spectrum, we run, as I mentioned previously, a database query where we go to a rotational spectroscopy database and find all known molecules with transitions that are nearby the observed line. In this case, there are 3 different molecules that match this criteria. then the first thing that we do for all of these three molecules is we determine their structural or chemical relevance to the source. And to do that, as I mentioned previously, we go to this chemical space surface, determine the weight at the points corresponding to those molecules, and this allows us to really quantify how chemically similar, how much it fits into the known chemical inventory in this source. Once we have this chemical relevance score for each molecule, we go ahead and run the spectroscopic checks. We take that score, scale it based on the frequency match of the catalog line to the observed line. We look for other transitions of that molecule in the data and see if the relative intensities match up. And finally, we end up with a score for each of the molecular candidates for that line in the spectrum. And if the top ranked molecule, the top scored molecule, if its score surpasses some predefined or pre-optimized threshold values, what we can do is we can confidently assign that line in the spectrum to the molecule in question. And what we also do is we build up a peak of weight at that location on the chemical space surface. Now that we know that molecule is present in the data, we can add it to our bank of information of the molecules present in the source. And then as we go on to the next line, we'll have a more informed chemical space surface. So as we go line by line, assigning more and more molecules, we really start to build up a very informed understanding of the regions of chemical space occupied by this source, or by the molecules in this source. And just as an additional note, after every subsequent assignment of a molecule, and as we update the chemical space surface because of this, what we do is we iteratively recheck all of the previous stronger assignments just to make sure, well, now that we have this new piece of information, that it doesn't suggest that in the past we missed something or we had a misassignment. So this algorithm goes line by line, strongest to weakest, until every single line, to the best of our ability, is assigned. So, yeah, go ahead.
Anyone has questions?
Yeah.
What rotational spectroscopy database are you using?
So we are combining, so the two main ones are the Jet Propulsion Laboratory, so JPL has a rotational spectroscopy database, and then the other one is CDMS, the Cologne group in Germany has a really nice database, and then just some molecules that have been studied in our various labs. We have catalogs, like local catalog fields as well. I have not, but something to look into. Yes. Yes.
You're mentioning how you had new particles, which already kind of have this weight regime figured out to some extent. For the first few particles that you're adding, is that still being done by hand?
No, it's all done by computer.
So like even when you have absolutely no idea when the weight is initially just off completely flat.
Great
and you're looking at that.
Yeah, so that is, as I said, the spectroscopic checks are not super like 100% robust, but for the first couple of molecules, we rely on those solely. So we still can, even if we don't have any understanding of how chemically relevant the molecule might be, we still can check whether the spectroscopy makes sense. And generally, we'll still have a top ranked molecule from that.
And this is a bit different question.
Yeah.
For that like similarity score, so it was trained on some large set of molecules, whatever the network to embed this into a vector space. Is that like, is the set of molecules it was trained on, is that the like a super set of all the molecules that you were looking at, or are there any things that you're trying to like generalize where like out of distribution of the initial training?
So the embedding model was trained irrespective to the data sets that were actually running. But not I guess that's not exactly true. So it was just like millions of molecules from PubChem and whatnot with certain filters to make sure that they're more acrochemically relevant. So certainly all of these molecules that we're assigning would have been in that training for the embedder, but there's millions of molecules and we're assigning them for the computer.
More questions? I do have one, sorry. I am still a little bit lost, sorry.
Yeah,
that
yeah.
I'm slow with, so can you go to the previous slide,
Yeah.
please? So,
This one?
yeah, I think this one should be good. Sorry, so you train the encoder on spectra of individual molecular species, or do you train it on combinations of several species?
So the encoder, so the actual embedder model is just for molecular similarity. The actual spectroscopic analysis is done, and the reason I didn't talk about it much is it isn't really anything machine learning based. We have spectral simulation softwares, and then pretty much through the through the comparison or the overlap of the simulated spectra with the observed spectra, we can sort of score it through that.
Oh, yeah, your input are just these tokens.
Just exactly,
Right,
it's
okay,
just so,
okay.
so what we're adding here, so the spectroscopic check. Or the ability to compare a simulated spectra with an observed spectra was something that had been done in the past. But the key here was that oftentimes, just because of overlapping spectra, things like that, it wasn't always super robust or easy to automate in the assignment of these pretty dense spectra based on the spectroscopy alone. However, things that weren't factored into previous automated assignment algorithms was the ability to say, does it really make sense that this molecule, if we have two molecular candidates, does it make sense that it's this one over this one based on what we've seen before, based on the homogeneity of the chemical inventory of that interstellar source?
Okay, so the way you construct that probability sort of landscape is by doing what exactly? So each point in that UMAP representation will correspond to one of these tokenized molecules.
Exactly.
So given that you have, and I understand it's initially just flat.
Yeah.
What's the process to actually build that landscape there? Can you explain
So
that?
yeah, it's for every new molecule that we assign, we simply just add a Gaussian peak of weight at that location on the chemical space surface. And the parameters of those Gaussian peaks were optimized just through a pretty simple hyperparameter search. But it's going by the concept of similar molecules are generally observed in these regions of interstellar space, and similar molecules are also nearby using these embedding models. You're able to quantify similarity between molecules.
Okay.
So by adding that peak, you're building a weight around that specific molecule. So similar molecules, nearby molecules on this chemical space will be highly weighted as well. So as you go line by line and assign more and more molecules, you have this superposition of a lot of peaks where it will just be sort of like a continuous surface of how likely any new molecule would be based on what we've seen before.
I think that's more clear.
Sorry, yeah, I didn't make that clear. All right, yeah, so I'll talk about some results on the assignment part of this algorithm, and then I'll get into the second portion, which is predicting the carriers of the unidentified transitions. So we've run this algorithm on the observations of TMC1, which is that cold molecular cloud. You can see the spectrum here on the screen, and here I put a green dot on all of the lines that were confidently assigned to a molecular carrier by the algorithm, and a red dot on those that remain unassigned. So there were not sufficient evidence to assign a molecular carrier to any of those lines with a red dot. They're typically these really weak transitions. But ultimately, our algorithm found 438 lines at 5 sigma strength, so 5 times the noise level, and was able to assign 425 of these to 47 unique molecular species. So there's only about 13 remaining unidentified transitions. And as I mentioned, with these really dense line surveys, if we were to manually cross-reference the rotational spectroscopy for every single line, it would have been a really painstaking process that probably took several weeks. The assignment part alone only took about a minute though. So it's a good efficiency increase for what we were able to do previously. And just to show the molecules that were assigned by the algorithm, I see all 47 here on the screen. And the good news is that all of these molecules are known to be present based on the previous literature in TMC1. So there are no false positive assignments, meaning that it's not assigning a line to a molecule that's not actually present in the data. And then the last thing I want to point out here is that there were 9 additional molecules that, based on how the algorithm considers spectroscopy alone, as I was talking about, is not always super robust. There were 9 additional molecules that actually aren't present in the data that would have been assigned if we didn't consider the structural relevance. So those 9 molecules just weren't chemically relevant enough or chemically similar enough on that chemical space surface to the known molecular inventory to be assigned. And as a result, that additional heuristic saved 9 additional false positive misassignments, which would have been about a 20% false positive rate, which just wouldn't have been a super useful algorithm at that point. So you can see here that adding in that consideration of structural relevance on top of the spectroscopic checks is really helping to get these accurate assignments in a way that we can fully automate. So as I mentioned, however, there are remaining 13 or so lines in this data set that were unable to be assigned by our algorithm. There wasn't enough evidence to assign them to a molecule for which a known rotational spectrum has been measured. And in fact, TMC1 is a really well-defined chemical inventory. So if we ran this on other data sets, it's likely there'd be a much larger proportion of these unidentified transitions. So the whole premise that we're going off of here is that molecules that are nearby or are highly weighted on this chemical space surface are strong candidates to be present in this data or in this interstellar source. So what we can go ahead and do is sample a lot of the highly weighted points on the chemical space surface, try to determine which molecules those could correspond to, and then go and say that those may be strong candidates to be the carriers of these unidentified transitions and run some follow-up experiments or calculations. But the challenge here is we have to figure out what molecule these are. If you remember when I talked about the molecular embedding model, it creates dense vector representations of molecules. It's unfortunately not creating like one-hot encoded vectors where it's a 1 if there's this functional group or a 1 if there's another functional group. It's not creating these directly interpretable vector representations of molecules. So that's exactly what we have to do. Our next goal is to train some sort of model to actually extract specific structural information or chemical information from these VIC-A embedding representations. So more specifically, what we've done is we've trained just a very simple feed-forward neural network model that takes as an input these VIC-A vector representations. I'm just showing it in 2 dimensions here, but it's the 32-dimensional vector, and outputting some probabilistic prediction that every selfies token in our dictionary would be present in the molecule. So in order to do this, we had to build up a large training set of molecules. And for this, we just randomly generated well over 1 million molecules by randomly sampling from our dictionary of selfies tokens, concatenating them into molecular representations, running them through the VIC-A embedding model. And that's our input to our model that we're trying to train. At the same time, for every molecule, we also produce a one-hot encoded vector denoting which selfies tokens are present in that molecule. So just as the example on the screen, HC9N, the selfies tokens that are used to construct that molecule are C, the triple bonded N, and the triple bonded C. So as a result, when we're training this model, the input will be the VIC-A vector representation of that molecule, whereas the output is this one-hot encoded vector denoting the actual specific selfies tokens that are in that molecule. So the overall goal here is that we're learning the specific chemical makeup of the molecules in each of these regions of chemical space. As a result, if we sampled a point around that of HC9N, the model would learn to predict that there are these selfies tokens in this molecule that make up these long chain unsaturated carbon species. Whereas if we sampled a point around indene, this polycyclic molecule, it would hopefully be outputting that there's a high probability of these ringed selfies tokens and able therefore to create these cyclic molecules. So ultimately, we went through the process of training this model and we're able to get quite good results at predicting the correct selfies tokens. And with that trained model, we could go ahead and try to predict or generate high probability candidates to be the molecular carriers of the unidentified transitions in this source. And in order to do this, the first thing that we want to do, as I mentioned previously, is sample a lot of highly weighted points on this chemical space surface. Then with the VIC-A representations of those highly weighted points, we run them through the model that we had just trained. And that gives us a probabilistic output vector that provides the likelihood that each selfies token in our dictionary would be present in a molecule in that region of chemical space. And now that we have that output vector, we can go ahead and actually start generating molecules that might be strong candidates here. And in order to do this, the first thing that we have to do is sample a molecular size. So sample the number of selfies tokens that we will use to build up that molecule. Say for this first example, we sample a molecular size of 3 selfies tokens. Well, we'll then go ahead and start probabilistically sampling the selfies tokens from that outputted vector based on their probability, 1 by 1. So here we first sample the C token, it's the most likely from that output vector. We then sample an oxygen and finally another carbon, and that provides the molecule dimethyl ether. We can then go ahead and start making another molecule. Say we sample a molecular size of 4 selfies tokens, go ahead and sequentially sample from that output vector based on the probability, and we ultimately end up with a molecule like glycolaldehyde. So we can go through this process tons and tons of times with every point that we sample on our chemical space surface to build up tens of thousands of different molecules. But unfortunately, it's not super useful to just provide a user with tens of thousands of different molecular candidates. That's way too much data to parse through. So we want to not only generate a ton of molecules, but we want to rank them as well. And in order to do that, for all of these generated species, we run them through the VIC-Day embedding model and then determine their weight on the chemical space surface following the assignment process. So we not only are generating these molecules, but we're ranking them based on their chemical relevance of what we know about this interstellar source. So just to get into some results on the molecular prediction part, ultimately on our TMC1 data, we generated 100,000 molecules, and that includes duplicates. So if you could think this process of sequentially sampling selfies tokens, you can end up with the same result a ton of times. So that 100,000 molecules, there's a lot of duplicates in there. But one exciting result is that over 14,000 of those generated 100,000 molecules were actually one of the 47 molecules that were assigned in our data set. And the reason that's exciting is because it suggests that this model that we've trained to predict selfies tokens is working pretty well. It's clear that if we're just creating the same molecules that we use to build up that chemical space surface, well, it's learning the correct selfies tokens that are represented to those regions of chemical space and therefore able to produce pretty relevant molecules. Just to show some more results, here you can see the top 100 ranked molecules from our algorithm following the assignment of the molecules in TMC1. And just flipping back to the last slide, you can see that they're visually at least very similar to those that were assigned in our data set. It's learning to predict or generate these very unsaturated alkyne containing carbon chain molecules. And I won't walk through every single one of these generated molecules out of time restraints. However, as I mentioned, we assigned 47 molecules in our data set. However, the known molecular inventory of TMC1 is quite a bit larger than that. There's a lot of known molecules that are either assigned via lines that aren't at 5 sigma strength, which is what we restricted our algorithm to, or those from other line surveys, which we didn't analyze in this work. However, the molecules that I will highlight here, including the top ranked generated molecule, are actually molecules that are known to be present in TMC1. So that's a pretty exciting result because it's showing that, in fact, that this method is able to produce very realistic candidates because it's actually predicting or generating molecules that are in fact present in this interstellar source, suggesting that the other ones could potentially be strong candidates as well. So while that's working pretty well, there's certain things that aren't working well at all. And the main issue that we're currently having with this method is the terminal heteroatoms in these generated molecules. So this means the non-carbon atoms at the end of these molecules, you can see them all here, they're oftentimes not in their correct form. So here I've circled all of the molecules that are predicted with NH2 groups. Well, anytime that we detect a molecule in TMC1 with a terminal nitrogen, it pretty much always has a CN group. So this C triple bonded end here, we pretty much never observe molecules in this NH2 form, with this NH2 terminal heteroatom. So what's going on here? Well, it's just the issue is that the way that we're sampling these selfies tokens sequentially isn't super robust. And just to show an example, say we sequentially sample these 4 selfies tokens to create a certain molecule. And here we are sampling this terminal heteroatom in its unsaturated form. So this double bonded oxygen is akin to like a triple bonded nitrogen. It's in its unsaturated form. However, when we sample it in this order, we end up with a carbon here with 5 bonds. We create a chemically invalid molecule. Unfortunately, just by sampling based on the probability vector that we're producing, we're not bound to create chemically valid molecules. And that's why we rely on the selfies, that Python package that has a decoder, which takes a chemically invalid molecule like this from its selfie string and decodes it into a smile of strings that would be chemically valid. But oftentimes, in order to do this, it changes the bond order. So you can see here, we generate this invalid molecule with an unsaturated terminal heteroatom, and the decoder, in order to make it a valid molecule, turns that double bond into a single bond. So unfortunately, the only way that we're going to be generating, using our current approach, these unsaturated terminal heteroatoms is if just fortuitously, all of the selfies tokens that we sampled prior to that allow that to be chemically valid, which unfortunately is just a pretty unlikely thing to happen. And that's why we're producing way, way more of these saturated, say like NH2 or OH groups instead of CN or carbonyl groups in this case. So what can we do to fix this? So our next steps moving forward on this project is we want to retrain that model that I talked about that we previously had trained to predict selfies tokens, but we want to now train it to actually predict molecular subgroups. So we're not just sequentially sampling selfies tokens anymore, but we're restricting when we're building up these molecules to maintain that local connectivity. Say that we're generating or we're sampling a CN group. If we sample the CN group as a whole, we're restricting it to maintain that connectivity, and it won't be able to be decoded into a way that it turns that unsaturated heteroatom into like an NH2 group. So that's really the work that we're going to try to do moving forward, as well as just applying this process to way more sources and continuing to improve both the assignment and the prediction. But with that, that's the bulk of the work I've done so far, and I'd be happy to take any questions you may have. So thank you.
Very interesting application of encoders and neural networks to chemistry. Any questions for Zach?
So, and I don't know much about this. So all the sources are very close, right? Or have known distance speed, I guess, because otherwise there would be like uncertainty.
Yeah, so actually a part that I didn't actually mention, the first part of this algorithm determines VOSRs and temperatures. So that impacts obviously the line frequency, the VOSR, and the temperature, the relative intensity, so things that are factored in. That's the first step of the algorithm that I didn't have time to really talk through. So before we get into the assignment, the first thing we need to do is determine temperature, VOSR, and alignment.
So it can be used for farm or with uncertain is
Yeah, I much more of a chemist than an astronomer. But so I'm generally using other people's observational data. So that's a good question. I'm not sure I have a perfect answer.
Thanks. Anyone else? Anyone on Zoom? So do I understand the problem you're having that you sort of explained at the end? That you are sometimes generating a token here that sort of makes sense in terms of the training data, but chemically it's not possible just because you cannot have, you don't have enough bonds in the carbon atom. I guess I'm trying to point out as to whether that's an issue with the GRU architecture. If the way in which you've learned that sequence does not incorporate any, information about the full relationship between these tokens, which brings me back again to the question I asked earlier as to whether someone would dare to replicate this method with a transformer that might be able to better associate relations between different elements of this tokenized version of the molecules. But I don't know, you seem to have a plan, so I'm not suggesting you that.
No, absolutely.
Yeah.
I think that the process of just sequentially sampling these selfies tokens could be done in a much more informed way. The way I'm doing it right now is quite a bit lazier. So I think just with more thought and a more robust way that actually considers that chemical validity, we could also get around this issue. Yeah.
Well, thanks very much.
Oh, sorry, we did have a question online.
Yes.
Harshal, you want to try to unmute yourself?
Yes. Hi, thanks. Can you hear me, Zach?
Yeah.
Oh, excellent. Thanks for the very nice talk. It's very nice work. I was wondering, like, what so your TMC1 assignments using this approach seem to be you had a pretty high success rate there, like 425 lines out of 438. So what was the I was wondering like how that was so successful, like what went into it, and you may have already explained this, but I was just curious, like what determined like assignment, and it was pretty impressive that you could assign it in just over a minute. Uh, so I was curious about what you attribute that success rate to.
Yeah, absolutely. So the assignment works in a way that we pretty much just first run a very simple peak finder on the data. So we're on the Gotham data, on the observations we have here. And for all those lines, we then query CDMS, JPL, the other databases that you would know for rotational spectroscopy for all known rotational transitions sufficiently nearby based on the line width of each of these lines. We then go ahead and using MolSim, so Brett's code, we simulate the spectrum and see whether the spectrum matches what we observe, whether like there are unaccounted for lines in the simulated spectrum, and whether it just matches up well enough with the observations. And then we also take into account how well that molecule fits into the data set. Because as you obviously know, Harshal, the molecules are pretty similar that we're detecting in TMC1. It's these cyanopolyions, as well as these long chain, maybe radical species. You're not getting like in high abundance these hot core species, these really saturated oxygen molecules. So using that consideration of structural relevance, we're also able to consider for those molecules, does the spectroscopy match, but also how chemically relevant it is to that interstellar source. And that's how we provide a score for each of those molecular candidates and assign it based on that.
I see. Yeah, no, thank you. And I'd be curious to learn more about this offline. The second, may I ask another question?
Go ahead.
Folks. So your comment reminded me of another. I was wondering, why you started with a really hot core in the first place, and I suspect I don't know what the motivation was for that, because so I'd be interested in learning about that as well. Like, what was your motivation? Was it simply because of the larger complexity of species that you expect to find in these, and you could test your approach on a more complex source than TMC1?
Absolutely. So it's a twofold answer there. The first one is that we want to make sure that this method is able to work on both TMC1 and a hot core, just to make sure that it's working for different source types and different chemical inventory types. Second of all, and this is a little bit of a sillier answer, IRAS 16293b is just a source that I've worked on a lot in my PhD work. It has a really well-defined chemical inventory, has really nice observations done. So it's kind of like we had the data on hand, we had all of that analysis already done, so we could test our algorithm without having to manually first assign all of those lines to see how well it's matching up with the automated approach.
Okay, great. Thank you so much.
Thank you. And thank you very much again. Zach is for local to us in Boston. Sometimes he might even show up here at CFA. So if anyone's interested in reaching out, please let me know. I'll be in contact. Thank you very much.